In this episode of Toxicity Tests, we're going to look at phototoxicity. For my definition of phototoxicity, I'm going to look at the Wikipedia site. This is the first time I've used this site. I'm a little bit hesitant to do so because I don't know its standing really in the academic world. When I was in the academic world, it didn't exist, so I don't know if you're allowed to source it or not, quote it or not. But anyway, it's got just a very nice simple definition of phototoxicity. The proviso is in the rectangular box there. It says this section does not cite any references or sources, so everything here has to be checked. But the definition here is a phototoxic substance is a chemical compound which becomes toxic when exposed to light. And at its simplest degree, I think this is what phototoxicity is about. Under the definition it reads, and you should check some of the pronunciations here a little bit as well. It reads some medicines, tetracycline, antibiotics, sulfonamides, amiodarone, quinolones. I think I pronounced those correctly. They can be phototoxic, and then it goes on. Many cold-pressed citrus essential oils, such as bergamot oil, some plant juices, parsley and hogweed, can be phototoxic. And under others, it says sorelin, which I'm not sure what that is. But you can see that quite a lot of things can be phototoxic. So anyway, that's what we're going to look at in this episode. This slide is from the Alltox website, and you can see that there are two validated non-animal alternatives to the animal tests. One is called 3T3 neutral red uptake phototoxicity test, and the other is called 3T3 neutral red uptake phototoxicity test application to UV filter chemicals. And they have got full OECD test guidance. Test guidance 432, that is the number of the test. Okay, let's have a look at the next slide now. If you look at paragraph 2, it describes the test, and it reads, OECD TG 432 describes the protocol for conducting the 3T3 NRU phototoxicity test. The assay measures the viability of mouse BALB/C 3T3 cells following their exposure to a chemical in the presence and absence of light. The paragraph continues, cytotoxicity, that's cell death, causes a reduction in the uptake of neutral red, that's a dye, by the cells. So that's how they're measuring this. The concentration dependent reduction in dye uptake 24 hours following treatment is used as the assay endpoint. Put simply, if a substance is phototoxic and therefore causes cytotoxicity, you will see this because there will be a reduction in the uptake of neutral red dye by the cells. So therefore you'll be able to classify it as phototoxic. The third paragraph talks about a totally different test. It reads, a test battery consisting of the yeast growth inhibition phototoxicity assay and the red blood cell photohemolysis assay has undergone an independent peer review by the Japanese Center for the Validation of Alternative Methods, and its acceptance by the Japanese Center for the Validation of Alternative Methods Regulatory Acceptance Board is pending. So let's go and have a quick look at that site to see what they're doing. Before that, I suppose we should have a quick look at how the Alltox website defines phototoxicity, and it's in the fourth paragraph. It reads, phototoxicity is defined as a toxic response from a substance applied to the body which is either elicited or increased after subsequent exposure to light, or that is induced by skin irradiation after systemic administration of a substance. So they're talking about sun creams and creams, the dermal applications really here. So it's slightly limited in its definition compared to the other one, I thought. But yes, they're talking about exposure to light and also skin irradiation, maybe the irradiation from light as well. Anyway, let's go to the Japanese site now. So here we are at the Japanese Centre for the Validation of Alternative Methods. Obviously this is the equivalent of the European Centre for the Validation of Alternative Methods and the US Centre for the Validation of Alternative Methods, which is obviously called the Interagency Coordinating Committee on Alternative Methods, if I remember correctly. So these are the three big alternative sites, the validation alternative methods organisations in this field of toxicology. So if we look here, we can see that yes, it's got a skin sensitization test and the corrosive test that we've looked at, and in the middle it's got phototoxicity, and it kind of gives timelines on their progress. So looking at the phototoxicity test timeline, we can see it goes through several stages. The validation study seems to be the first stage, then it's peer review, regulatory acceptance, recommendation to government, and then OECD acceptance, full test guidance acceptance, I suppose, which seems to be the end point, the highest status a test can get. So looking at the yeast growth inhibition phototoxicity assay and the red blood cell photohemolysis assay, we can see that the validation study started in January 08. By May 09 the peer review had taken place, and by January 2010 it seems to have got regulatory acceptance. Under the yeast growth inhibition test, you can see that the Japanese Centre for the Validation of Alternative Methods are looking at another test called the ROS assay, and its status is that it's going to start the whole validation process soon, it says. And it's got a purple dot, or a purple dot to signify that fact. Okay, if we look at a couple more of the slides from the Japanese Centre for the Validation of Alternative Methods, we can see what else they're working on and what else has got full approval. This slide shows tests for skin sensitization, which we've looked at, skin irritation and ocular irritation tests, all of which we've had a look at. You can see the timelines here. The orange dot seems to be the final dot, the highest status dot. That's the OECD test guidance dot. So you can see that there's a blue dot which signifies that cytotoxicity. With regards to ocular irritation test, that seems to indicate that it's just beginning. There's one that's revised there. Maybe it got all that way and then they weren't sure about it, so they asked for other things to be looked at before they were willing to accept it as a full test. In this last slide we can quickly take a look at the other progress of the other tests that are taking place. For instance, we can see the endocrine disruptor screening assays. One of them, I think two of them actually have got full approval now, but it says number 459 has got full approval, and from that episode that we looked at endocrine disruptors, number 455 has also got full approval now. So you can see, like, the European Centre for the Validation of Alternative Methods, the US Interagency Coordinating Committee for the Validation of Alternative Methods, and this one, of course, the Japanese Centre for the Validation of Alternative Methods, look closely at the alternative methods that are coming from the scientists to see whether they stand up to scrutiny and can be passed as replacements or reductions to the animal tests. Here we're looking at the actual OECD test 432 in vitro 3T3 NRU phototoxicity test. In the introduction we can see where the Alltox got their definition for phototoxicity. It reads: phototoxicity is defined as a toxic response from a substance applied to the body which is either elicited or increased after subsequent exposure to light. So that's pretty much the same as the Alltox definition. In the second paragraph it reads: the in vitro 3T3 NRU phototoxicity test is used to identify the phototoxicity potential of a test substance induced by the excited chemical after exposure to light. The test evaluates cytotoxicity by the relative reduction in viability of cells exposed to the chemical in the presence versus absence of light. Substances identified by this test are likely to be phototoxic in vivo. I'm just interested in paragraph 2 of this slide. It reads: the reliability and relevance of the in vitro 3T3 NRU phototoxicity test was recently evaluated. The in vitro 3T3 NRU phototoxicity test was shown to be predictive of acute phototoxicity effects in animals and humans in vivo. The test is not designed to predict other adverse effects that may arise from combined action of a chemical and light. For example, it does not address photogenotoxicity, photoallergy or photocarcinogenicity, nor does it allow an assessment of phototoxic potency. In addition, the test has not been designed to address indirect mechanisms of phototoxicity, effects of metabolites of the test substance, or effects of mixtures. So this OECD test seems to be specifically looking at the toxic response from a substance as applied to the body, like I said, such as a sun cream or any other cream, say for eczema, hydrocortisone cream for instance. So it's not really looking at a substance that is inadvertently left on the windowsill in direct sunlight and then becomes toxic. Other tests will look at that kind of toxicity, say genotoxicity or carcinogenicity tests. So I just want to clear that up, that we are looking at a specific area of phototoxicity here and not a comprehensive overview of the whole topic. I'm just going to read the last sentence of this slide. It says, to predict the phototoxic potential, the concentration responses obtained in the presence and in the absence of irradiation are compared, usually at the IC50 level, i.e. the concentration reducing cell viability to 50% compared to the untreated controls. Obviously your untreated controls are going to be kept in the dark. In this slide we're looking at the cells, and it says a permanent mouse fibroblast cell line, clone 31, either from the American type culture collection or from the European collection of cell cultures in Salisbury, Wiltshire, was used in the validation study, and therefore it is recommended that cells be obtained from a well-qualified cell depository. So what I understand here, a permanent mouse fibroblast cell line, I imagine that is a continuous cell line, so I don't think any animals actually have to be killed in this test, which is really good. And it just shows you that you have to get quality cell lines to be able to compare like with like around the world. Okay, this is the last slide from the OECD, and it's about interpretation of results. It reads, based on the validation study, a test substance with a PIF, which stands for photoirritation factor, less than 2, or an MPE, a mean photo effect, that stands for less than 0.1, predicts no phototoxicity, a PIF greater than 2 and less than 5, or an MPE greater than 0.1 and less than 0.15 predicts probable phototoxicity, and a PIF greater than 5 or an MPE greater than 0.15 predicts phototoxicity. So just 3 more slides to go, and they're all from the Canadian Council on Animal Care. In this slide we can see the conventional test method in the left hand column, in vivo mammalian phototoxicity test. However, I have no idea what the test actually is, because it doesn't seem to have an OECD test guidance number. Anyway, let's have a look. So we've looked at the in vitro 3T3 neutral red uptake phototoxicity test. And we're going to now look at the validation status and the regulatory status. And there's a very strange regulatory status to this test, which I'll show you in the very last slide. I'm a little bit conscious of being repetitive with this slide and therefore boring, which is the last thing that I'd ever want to be, but it gives a very good, clear description of the test, the in vitro 3T3 neutral red uptake phototoxicity test. It reads, this test predicts acute phototoxicity effects in animals and humans in vivo. The viability of mouse 3T3 cells is determined following their exposure to a test compound in the presence and absence of stimulated solar light. So they're not actually putting it in front of the sun on a windowsill, they're using lights in the laboratory. Cytotoxicity is measured as a reduction in the uptake and binding of neutral red dye of the cells. So let's have a look at the very last slide now to find out about this unusual regulatory advice. Here it is then, under regulatory status, OECD TG 432 brackets 2004, EU, accepted by European Commission as method B.41 in Annex 5 of EU Council Directive 67/548/EEC. This test must be used to determine phototoxic effects of chemicals, and animal tests are prohibited for this purpose in all EU member states. Now that is extremely unusual that they've made this decision, and if they can make that decision once, they can make it again. So I find that incredibly encouraging. I don't know why they say that the animal tests are prohibited for this purpose of testing phototoxicity, but the fact that they have signifies hope for the future, in my opinion.